In this video, you will see how to run a ready-to-use Novex ELISA kit. The kits come with a pre-coated 96 well plate with capture antibody at the bottom of each well, and all the necessary buffers and standards for running the ELISA. Open the foil pouch to remove the plate and remove any 8-well strips that will not be needed for the run. Unused strips can be rebagged and stored at 2 to 8 degrees Celsius for future use. To prepare wash buffer, you will need to allow the wash buffer concentrate to reach room temperature and mix to dissolve any precipitated salts. Dilute 1 volume buffer into 24 volumes of deionized water. Label as working wash buffer. 400 microliters of wash buffer per well or 3.2 mils per strip of wash buffer is required for each washing step. Reconstitute 1 vial of standard with standard diluent buffer. Swirl gently and let the standard sit for 10 minutes at room temperature to ensure complete reconstitution. Label vials and perform serial dilutions of the standard, adding appropriate amounts of the standard into each tube of standard diluent buffer. Add standards, controls, and natural samples to appropriate wells. Note that samples may need to be diluted. Allow the antigen to incubate. Next, you will need to wash unbound antigen from the wells. For sequential assays, antigen incubation time is 2 hours. For co-incubation assays, antigen incubation time is 1 hour. To wash, either manually wash or use an automated plate wash. To wash manually, completely aspirate the liquid from all wells, taking care not to scratch the inside of the well. Or, invert the plate over a sink and shake the fluid out. Fill the well with at least 400 microliters of wash buffer. Allow the wash buffer to stand for 15 to 30 seconds. Then decant again. Repeat at least 2 more times. After washing, invert the plate and tap dry on absorbent paper towels. This washing step will be repeated throughout the protocol. You are now ready to add the next component, the biotinylated detection antibody. The detection antibody will bind to a different site on the antigen to create a sandwich. For sequential assays, incubation time is 1 hour. Please see the technical datasheet for more details on the assay format. After washing away unbound detection antibody, add streptavidin horseradish peroxidase. In this step, multiple biotin streptavidin binding sites could occur on each secondary antibody molecule, leading to an enzymatic amplification of the signal. For sequential assays, incubation time is 30 minutes. Wash the plate as above to remove any excess streptavidin horseradish peroxidase. Once excess enzymes are removed by washing, add chromogenic substrate. The intensity of color developed is proportional to the antigen concentration of the sample. Incubate the plate in the dark at room temperature for 30 minutes. To stop the enzymatic reaction, add 100 microliters of stop solution. The solution will turn yellow. Read the plate by measuring absorbance at A450 nanometers. The concentration can be calculated from the standard curve.